pollen pistil interaction. So far what we have studied is that is something related to pollination, right? Now pollination is a process where anther is now transferred, pollen grain is transferred from anther to the surface of the stigma. Now the question is, what happens when the pollen grain lands on the stigmatic surface? That process we will study in pollen pistil interaction. There are some pictures given here. Imagine hundreds of pollen grains, they deposit on the stigmatic surface. See, in second PUC in lab we'll study some experiments where we will collect the stigma from certain plants, and those stigma are boiled, and with the help of cotton blue stain they are squashed, and those stigma are observed under the compound microscope. What you can see on the surface of the stigma, there are a lot of hair-like structure and hundreds of pollen grains. Some of them they germinated and pollen tubes are clearly observed in the style. So this is how we can observe the germination of pollen grains on the stigmatic surface. Now the question is, how this pollen grain will undergo pollen tube formation, or it undergo germination? There are certain type of chemicals which allow the pollen grains to germinate, which is somewhat similar to 10% sucrose solution. In laboratory we use 10% sucrose solution to germinate the pollen grain and observe the pollen tube formation from the germ pore. Here on the stigmatic surface, again somewhat similar to this sucrose solution will help the pollen grains to germinate. First and the foremost thing, the pollination will not ensure the fertilization, because the pollen grain may come from any plant. Pollen grain may not be come from the same plant and land on the stigmatic surface. So the first thing need to be done is to find out whether the pollen grain belongs to same species or different species. So there are some genetic mechanism with the help of that plant will detect the pollen from the same plant or same species, then only it allow the pollen grains to germinate. Hundreds of pollen grains are deposited on the stigmatic surface, and they starts to produce the pollen tube, and the pollen grain which have the ability to produce the pollen tube faster reaches the ovary where ovules are present. Now pollen tube come in contact with the micropylar region of the ovule and releases its male gametes. Now question comes, how many male gametes are present? Already we studied that male gametes are 2, but sometimes what happens, in 60% of the plants pollen grains are shed at the 2-celled stage. Now when they are released at the 2-celled stage, one of the nucleus which is present in the vegetative cell help in guiding the pollen tube formation, and one of the nucleus which is present in the generative cell acts as the male gamete. Now this generative cell, while moving along with this pollen tube, in the half the way they undergo mitosis and produces another male gamete. So when the time it reaches the micropylar region, there will be 2 male gametes. Okay, the rest of the 40% of the plants, they already produce the 2 male gametes before the shed. So pollen grains are released in 3-celled stage in those 40% of the plants. Okay, so same thing is explained here. First they will plant stigma will find out whether it is a compatible pollen or incompatible pollen, and then allow it to germinate. Okay, so when pollen tubes are formed, they will pass through the tissue of the style and then reaches the ovule. Here you can see this is the pollen tube. Now it reached the micropylar region. Here the embryo sac is present. Now embryo sac is consisting of egg apparatus, polar nuclei and antipodal cells. Okay, so the region where the pollen tube enters, that is the micropylar region, and the opposite region is the chalaza. So let us see what and all the explanation is given here. Pollination does not guarantee the transfer of right type of pollen. Why they are saying this? Because the insect which is visiting one flower, you cannot say it visit only one species. So it has visited several species. Its body is having different pollen grains. So there is high chance that the pollen of desirable plant may deposit on the stigmatic surface. Often, pollen of the wrong type, either from other species or from the same plant, also land on the stigma. Okay, so there is a reason to give if it is self-incompatible, because if the plant is encouraging outbreeding device or cross-pollination, in that case, suppose insect is carrying the pollen from the same flower, is also a problem for the plant. So at that time, pistil has the ability to recognize the pollen, whether it is of the right type, or you can say compatible type, or the wrong type. If the right type, if it is right type, the pistil accept the pollen and promote the post-pollination events that ends to fertilization. If the pollen is of the wrong type, the pistil rejects the pollen by preventing the pollen germination on the stigma or the pollen tube growth in the style. The wrong pollen grains are deposited on the surface of the stigma, then they are rejected. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of continuous dialogue between the pollen grain and the pistil. See how beautiful it is. We think we are brilliant. Just look at the plants. They have ability to recognize the pollen grains just on their surface, right? Yes. Now the dialogue is mediated by chemical components of the pollen interacting with those of the pistil. So it's the only recent years that botanists have been able to identify some of the pollen and pistil components and the interactions leading to the recognition followed by acceptance or rejection. So far, we tried hard to understand what type of interaction takes place between pollen grain and the stigmatic surface. Recently, scientists, they are able to find out the type of interaction taking place between pollen grain and the stigmatic surface. The pollen grains germinate on the stigma to produce pollen tube through one of the germ pore. One of the germ pore means what? Please do remember with this sentence, one pollen grain does not have one germ pore. They have multiple germ pores. Okay, but from the one germ pore, which is directly facing the stigmatic surface, it starts to produce the pollen tube. The contents of the pollen grain move into the pollen tube, and the pollen tube grow through the tissues of the stigma and style and reaches the ovary. Then pollen grains are shed at the two cell stage. It is just for recall they have given. Already I explained what happens when it is released in 2-cell stage and when it is released in 3-cell stage. When it is released in 2-cell stage, when it travels through the pollen tube, in the half the way it again undergo mitosis and produces the 2 male gametes. When the pollen grains are released in the 3-cell stage, 2 male gametes just follow the pollen tube and reaches the embryo sac. Now, pollen tube, after reaching the ovary, enters the ovule through the micropyle and enters the synergids through the filiform apparatus. This is very, very interesting actually. Just observe what happens when pollen tube reaches the micropylar region. The nucleus which is guiding this pollen tube reaches micropylar region and its function is over. Now there are 2 male gametes. Somebody has to guide them and help them to fuse with the egg cell and the polar nuclei. That function is done by filiform apparatus. Okay, so these synergids lie near to the egg cell. They have finger-like projections. These finger-like projections guide this male gametes, male nucleus towards the egg cell and polar nuclei. Now what happens? One of the male gamete fuses with the egg and forms syngamy. This is very important process, nothing but the fertilization. Now one more male nucleus fuses with the polar nuclei and forms the triple fusion. One is syngamy, another one is triple fusion. Here you observe 2 fertilization. One is fertilization of egg, another one is fertilization of polar nuclei. So the term is used double fertilization. And one more term is used, that is triple fusion, because polar nuclei is already diploid. Now it is fusing with the haploid male nucleus. The process is called triple fusion. So same thing is explained here. See, one of the experiment which we perform in the laboratory, that is given here. Take a pollen grain from different plants or any one of the plant, say for example impatiens balsam, or any of the nearby plant. You take the pollen grains like hibiscus, then take the pollen grain of a very young flower, that means the flower which bloom in the morning, and dust this pollen grain on the slide having 10% sucrose solution. Keep this slide for 20 to 30 minutes for observation. Then observe the slide. You see the pollen grains forming pollen tubes. Okay, so there are multiple germ pores. A small pollen tube will be formed in those areas, and one of the germ pore produces very long pollen tube. Okay. Now coming to one of the very important technique. See, artificial hybridization is one of the very important technique practiced long back to produce the hybrid plants. See, how to assemble desirable characters to get the best offsprings. Scientists, they search different ways and they come up with the idea. By knowing the knowledge of pollen pistil interaction, they just implemented those techniques in artificial hybridization. Suppose the plant is bisexual, produces androecium and gynoecium. A plant breeder, what he will do? He will take the plant, say for example pea plant, before the anthesis, before the flower opens, he take out the petals and he observe the stamens. With the help of a suction pump, a small suction pump, or with the help of needles, he remove all the male reproductive part, that is stamen. This process is called emasculation. Very important. In artificial hybridization, emasculation means what? Removal of stamen from the flower. Now once the stamens are removed, you are very sure that the self pollen cannot deposit on the stigmatic surface. Now, in order to make sure that the pollen from some other plant come and deposit on the stigmatic surface, what he will do? He will cover the flower with the help of very thin transparent polythene bag or any type of muslin cloth. Now, in the bagging process, the whole flower is covered. Next, what he will do? He will take the pollen grains from the desirable plant. Somewhere he observed one plant, it is producing very high yield with the disease resistance. So he collected those plant pollen grains, and here what he is growing is the type of plant which has drought resistance. So he want to include those two characters. So he dust those pollen grains on the stigmatic surface when the flower matures. Now this we call it as artificial hybridization technique. Artificially you transfer the pollen grain, and rebagging is done to avoid further contamination of the stigmatic surface. So this makes sure that the desirable pollen is transferred on the stigmatic surface and produced offsprings will be hybrids. Okay, but there is no guarantee that the produced offsprings will be hybrid, but there is high chance that produced offsprings will be hybrids. If the female parent produced unisexual flowers, there is no need for emasculation. Okay, suppose the flowers are unisexual, then there is no need of emasculation. Simply you take the pollen grain from the desirable plant and then transfer it to this flowers and rebag it. There is a high chance that you will get the hybrid plants. So this is how artificial hybridization is conducted to get the better plants or better offsprings. Now coming to double fertilization. See, so far we traveled so much with this sexual reproduction in flowering plants. We started from microsporogenesis, megasporogenesis, and after that pollination, in that kinds of pollination, then different modes of pollination, in that water pollination, wind pollination, insect pollination. See, after that pollen pistil interaction, all these process is nothing but pre-fertilization events. Now we will study fertilization. What happens when the male gamete come in contact with the female gamete, right? See, this is very interesting part. Already we discussed a little about this double fertilization. As I said, pollen tube, they germinate in the on the stigmatic surface and passes through the style and reaches the ovary, where in the locule so many ovules are present. So many pollen tubes, they reach the different ovules and they release the male gametes in the micropylar region, and these male gametes are directed by the filiform apparatus which is present on the synergids, and they guide these male gametes to reach the egg and polar nuclei. So one of the male gamete fuses with the egg and leads to syngamy. It makes sure that now fusion of male and female gametes leads to the formation of zygote. In other words, at the same time, another male gamete fuses with the polar nuclei, and we call it as triple fusion, and which makes sure that this polar nuclei after fertilization develops into endosperm. There is one important aspect here. Why there is a need of endosperm? And one more thing, endosperm formation takes place first and embryo development takes place later. Sometimes in competitive exam they particularly take these sentences. Okay, so the endosperm formation takes place first and embryo development takes place later. The reason is developing embryo make sure that there is sufficient food for its development. So endosperm development takes place first and then embryo development takes place. Now there are the high chance that the questions may ask based on the ploidy. Now tell me, what is the ploidy of endosperm? It is triploid because primary endosperm nucleus is already diploid. One more male gamete come in contact with it and formed a endosperm. Now the endosperm is 2 plus 1, that is 3. It is triploid. Endosperm is always triploid, and the zygote formed is diploid. Okay, though this process we call it as double fertilization. Fertilization of egg and fertilization of primary endosperm nucleus takes place simultaneously, and this process we call it as double fertilization. Okay. Now here in this picture you can see that the synergids after fertilization, they undergo degeneration. That means whatever the nutrient content is there, that will be consumed by developing zygote, and the endosperm nourishes the developing zygote, and zygote will develop into embryo. These antipodal cells also undergo degeneration. This zygote later develop into different stages of embryo that we will study in detail in embryo. Here you can see globular embryo, heart-shaped embryo, and a mature embryo where you can clearly observe the cotyledons. So this is definitely an example of dicotyledon. So in this post-fertilization structure and events, we study in detail about the endosperm, in detail about the zygote developing into embryo. So we'll study embryos. And then what happens to ovule? The fertilized ovule develop into seed. We study in detail. What happens to ovary? Ovary develops into fruit. That we will study in detail. Okay, so these are the aspects which we are going to study in post-fertilization structure and events. Let us see one by one. In that, first one is endosperm. Just now I said, what is endosperm actually? When the primary endosperm nucleus undergo fertilization with the help of male gametes, endosperms are formed. Now tell me whether endosperm is formed first or embryo development takes place first. Definitely endosperm formation takes place first. Now whether this endosperm is retained in the seed or it is completely consumed, or which is the form of endosperm we commonly observe. Hope you have tasted a tender coconut. What is this tender coconut actually? It is the free nuclear endosperm. This endosperm is formed continuously by multiplying its nucleus, which is not followed by the cell wall formation. So only after the maturation of coconut, you see a kernel which is white colored. where endosperm nucleus is now surrounded by the cell wall formation. Before that, it is all a nucleus, free nuclear endosperm. This endosperm sometimes completely consumed by the growing embryo. Such seeds are called non-endospermic seeds. So can you give me some example for non-endospermic seeds where endosperm is completely consumed by growing embryo? The best example is dicotyledons, like groundnut, like badam, like cashew nut, where completely this endosperm is consumed. If you cut open the seed and observe, there will be a groove which is empty, and at the base of that, the small embryo is observed. Can you tell me some example where endosperm is retained even after the seed formation? The best example is monocot seeds like wheat, rice, coconut, castor, where you observe the endosperm even after the complete embryo development, and in the seeds these endosperms are present. So based on that, we will classify them as albuminous seeds and non-albuminous seeds. Albuminous seeds means endosperm is present. Non-albuminous seeds means endosperm is completely consumed. Okay, so that is about the endosperm. Next part is about the embryo. Now the study of embryo is why so important. Whether the embryos are same in all the cases. In the case of angiosperms, no, that's not true. In angiosperm, we observe major two classifications, that is monocots and dicots. Now, monocots have one type of embryo and dicots have one type of embryo based on their cotyledons and some other structures that we will study in detail. Okay, just observe this picture. What happens to zygote? Zygote is diploid. As it starts to divide mitotically, the number of cells will increase, and initially it just form a mass of cells, and those mass of cells, they slowly start to differentiate, and first it forms the globular embryo. So this is the structure of globular embryo. This is the suspensor actually. This part actually attaches to what it attaches to micropylar region. Okay. Now this suspensor at the end, you can see here it is heart shaped, and finally it completely matures and forms the two cotyledons. Now the same structure is given here. Now when you observe this dicot embryo, where you can differentiate the hypocotyle region and epicotyle region. In the epicotyle region, you observe the plumule part and cotyledons. Actually this plumule, after germination of the seed, develop into shoot. In the hypocotyle region, you observe the tubular part. At the end, radicle is present. This radicle later develops into root. Okay, this root is covered by a protective layer that is called root cap, which is made up of calyptrogen. So the thing is, dicots, they have two cotyledons. Now my question is, what is this cotyledon? Why they are present in the embryo? And even in the seeds, you can see the cotyledons. Whenever you cut open the groundnut seed, you can see two cotyledons. Or any of chana dal or anything, you cut open the seeds, you observe two cotyledons. They are actually leaf of the embryo. Whenever this seed starts to germinate, it doesn't have a proper root system to support the plants, or it needs to develop the root system that requires the food. So those food is stored in the cotyledons, and with the help of cotyledons, first they establish well root system, and then with the help of the nutrients remained in the cotyledons, they produce the shoot system and then produces the leaves. So before the leaf formation, cotyledon acts as an embryonic leaf. Please do remember, this is the very important concept. Okay, hope you have just observed the dicotyledon embryo. Now observe the monocotyledonous embryo. There are a lot of changes in monocotyledonous embryo. Why the name monocotyledon? Because it has only one cotyledon. That one cotyledon is represented by the scutellum. Okay, scutellum represents the single cotyledon. Now, if you observe the embryo, this is the shoot part, nothing but shoot apex. You can say plumule. and that is covered by a protective layer called coleoptile. This was not observed in dicotyledonous embryo. Here plumule is covered by coleoptile and radicle is covered by coleorhizae. Okay, here you can see a small part called epiblast. Explanation is not given in textbook, but can you predict what is that epiblast and why it is given? See, it's just a ruminant part. Okay. Actually, if it grows and form one more cotyledon, it would have observed like a dicotyledonous seed. But in evolution, this part got reduced. Okay, it's a reduced cotyledon. And one cotyledon is very prominent, that we call it as scutellum, that acts as a single cotyledon in the case of monocotyledons. Hope this is very clear. Here shoot is covered by a protective layer called coleoptile and root is covered by a protective layer called coleorhizae. So this is the main difference between monocot embryo and dicot embryo. Next concept is seed. See, tell me, what is seed actually? Embryo is nothing but the zygote which develops and becomes embryo. Where it is present inside the seed. Now what is seed? Right? Now recollect the ovule. Inside the ovule, embryo sac is present. Now embryo sac has egg apparatus. Now egg after fertilization develop into embryo. What happens to the ovule? That same ovule after fertilization develop into seeds. Okay. Now, what is the importance of seeds in plants? Because the production of seeds makes sure that that plant can produce the next generation. Because in lower plants, we observed that the body of the plant itself undergo breakdown by fragmentation and each fragment grow into a new plant. Or there are certain mechanisms where the offset, the suckers or different types of structure produced in the vegetative body of the plant itself give rise to a next generation. But in sexual mode of reproduction, this is the mean for production of offsprings, seed. So please do remember that seed is nothing but fertilized ovule. Now, is there a connection between the number of ovules and number of seeds? Of course. The number of ovules get fertilized will develop into number of seeds. Okay. Now in angiosperm, seed is the final product of sexual reproduction and it is often described as a fertilized ovule. Seeds are formed inside the fruit. Now what is fruit actually? Fruit is nothing but the fertilized ovary. Inside that ovules are present, right? So ovules are fertilized and develop into seeds and ovaries are after fertilization develop into fruits. A seed typically consists of seed coats. How the seed coats are formed? Just observe the ovule. You can see the integuments. These integuments later develop into seed coat. That seed coat, inner seed coat is called testa and outer seed coat is called tegmen. The cotyledons of the embryo are simple structures, generally thick and swollen due to the storage of food reserves. The cotyledons, which is part of the embryo, which is mainly for storage of food. Mature seeds may become non-albuminous or X-albuminous. Why they become non-albuminous or X-albuminous? Because growing embryo, if it completely consumes the endosperm, then we use the term non-albuminous, X-albuminous or non-endospermic. All these terminologies are same. Non-albuminous seeds have no residual endosperm as it completely consumed during the embryo development. There are some exceptions also. Okay, albuminous seeds retain part of the endosperm as it not completely used during the embryo development. Like in the case of wheat, maize, and barley and castor, they retain the endosperm even in the seeds, and those seeds are called albuminous seeds or endospermic seeds. Okay. Occasionally, some seeds such as black pepper, this is one exception. Please do remember, very, very important example. Black pepper in beet, remnants of the nucellus are also persistent. Okay, the terminology used for this remnant nucellus or persistent nucellus is perisperm. Most of the time in competitive exam, they focus on this type of terminologies. What is the meaning of perisperm? Persistent nucellus. Persistent means what? Actually, after fertilization, nucellar cells should undergo degeneration. But even after the fertilization, in the seeds, you observe nucellar cells, and such persistent nucellus is called perisperm. Now, integuments of the ovule harden as tough protective seed coats. The micropyle remain as a small pore in the seed coat. If you observe any of the seeds, like you take the example of Kabul or Channa, observe its end. At one point there is a hole. That hole is nothing but what? Actually a micropylar region. Why it is present? If you soak the seed in the water, water can enter through the micropylar region and it helps in imbibing the water, and seed can swell, and this swelling help them to germinate. Okay, so the micropylar region will be persistent even in the seeds also. This facilitates the entry of oxygen and water into the seed during germination. As the seed mature, its water content reduced and seed becomes relatively dry to 10 to 15%. Now the question comes, why seed should undergo dry? See, most of the times on set of favorable conditions, plant will grow and they produce very beautiful vegetative structures, and they produce flowers, and flowers undergo pollination and seed setting will take place. When the time comes to seed setting, the plant will experience a unfavorable condition or harsh condition. In case the produced seeds, when they fall on the soil and immediately undergo germination and produces the plantlets, and plantlet starts to grow, the coming seasons are completely unfavorable, like it may be winter or it may be summer. So there is very less chance that this plant will survive and produces the next generation. So in most of the case, what happens, the produced seed will retain very less amount of water, 10 to 15%, and undergo dormancy. What is dormancy actually? It is a inactive viable time of the seed. Inactive viable means what? Seeds are viable. They have ability to germinate in onset of favorable condition. But for a time interval, it slow down all its physiological activity and undergone dormancy to make sure that this seed will be viable in the unfavorable condition, and onset of favorable condition they can germinate. Okay, so the general metabolic activity of the embryo slows down, and embryo may enter a state of inactivity called dormancy. This dormancy will vary. Okay, you cannot say seeds may be dormant for 3 months, seeds may be dormant for 4 months, or seeds may be dormant for 1 year. It varies from plant to plant. There are certain examples, you will be surprised. Some seeds are extracted from the place where these seeds are viable for thousands of years. Surprising, right? For thousands of years, they retain their viability. I will give you those examples. Okay, as the ovule mature into seed, the ovary develops into fruit, and the transformation of the ovule into seed and ovary into fruit proceeds simultaneously. It is not a separate process. First seed formation takes place, then fruit formation takes place. There is no such things. So these two, formation of seed as well as formation of fruit, takes place simultaneously. Wall of the ovary develops into a wall of the fruit called pericarp. In morphology, you have studied this. What is pericarp? Pericarp is present external to the endocarp, where mesocarp and epicarp is present. Okay. The fruit may be fleshy, as in guava, orange, mango, etc., or may be dry, as in groundnut and mustard. There are different types of fruits: fleshy fruits, dry dehiscent fruits. Okay, so some of the examples you studied in morphology of flowering plants. Many fruits have evolved mechanism for dispersal of seeds. So this is a really interesting type of mechanism. Have you seen impatiens balsam or balsam plant? If you just touch the mature fruit, the fruit will burst and releases the seeds to the environment. Okay, so the different plants have different techniques. You can see a pappus. During winter season, a small parachute like seed will fly around us. This is also a type of seed dispersal mechanism, parachute mechanism of seed dispersal. Okay. So is there any relationship between number of ovules in the ovary and the number of seeds in the fruit? Of course, there is a relationship, because the number of ovules present inside the ovary, the same thing after fertilization develop into seeds, and the ovary develops into fruit. Now, in most plants, by the time fruit develops from the ovary, the other floral parts degenerate and fall off. Why they have used the term most of the plants? There are certain examples. Let me tell you, in the case of brinjal, tomato, or in capsicum, the sepal will retain on the fruit also. That we call it as persistent sepals, right? So they have used the term most of the plants. Okay. However, in a few species such as apple, strawberry, cashew, the thalamus is also contribute to the fruit formation. Tell me, what if other than ovary forming a fruit, what we call it as true fruit or false fruit? Of course, it's a false fruit. Sometimes what happens, the ovary will not develop into fruit, instead thalamus will develop into fruit. So for that they have given some of the example like apple, strawberry and cashew. These are the best example for false fruits. Such fruits we call it as false fruits. Most fruits, however, develop only from the ovary and are called true fruits. You have hundreds of different examples for true fruits, where most of the times ovary develops into fruit after fertilization. Although in most of the species fruits are the result of fertilization, there are few in which fruits develop without fertilization. See, this is again an exception. Most of the times the fertilized ovary develops into fruit, but there is a chance where fruits are produced without fertilization, and those fruits are called parthenocarpic fruits. So in nature, you observe one of the best example, that is banana. Have you seen banana plant? Now tell me, if the ovary is directly developing into fruit without fertilization, what is the essentiality of producing flowers in banana plant? Stigma, style, petals, everything is just a waste. It just requires gynoecium, and that gynoecium develops into fruit. So that's why in banana you can see it produces very much large colored bracts. They are simply covering all the flowers, because flowers, they do not require any type of pollinators. Even though the insects, if they visit the flowers, they will get reward in the terms of nectars, but they don't require insects for pollination. Fruits are produced without fertilization, and that condition we call it as parthenocarpic fruits. Now the question comes, when the fruits are produced without seeds, it's very easy to eat. Can we induce this character in any of other plants so that we can produce a type of without seeds? Imagine a watermelon without seeds. Imagine grapes without seeds. Imagine orange without seeds. How easy to eat, right? So there are some practical applications where they use plant growth regulators like auxin to produce parthenocarpic fruits. So in market you will get seedless grapes, seedless orange. All these are what? Actually they used plant growth regulator like auxin to produce the seedless parthenocarpic fruits. Here are some examples. You can see this is the thalamus in the case of apple, and this is one monocot seed vertical section. Here you can see the endosperm. So the maize seed is an example for endospermic seed. This is the scutellum, single cotyledon, and here you can see coleoptile, plumule, radicle and coleorhizae. What is this pericarp actually? Proteinaceous layer, that is pericarp. Okay, so here is the strawberry. These are some of the example of embryo, and this is the cotyledon in the case of dicots. There are some questions which appeared from the these diagram based questions in NEET and CET that we will discuss in the later part of the topic. As they have sufficient food reserve, young seedlings are nourished until they are capable of photosynthesis on their own. That's why earlier in this topic I explained what is the role of cotyledons. They act as embryonic leaf. Okay, the hard seed coat provides protection to the young embryo. The being products of sexual reproduction, they generate new genetic combinations leading to variation. What is the main intention of sexual mode of reproduction? Producing offsprings having some variations. So what is this variation and why it is so important? Variation leads to a better chance to the offsprings to survive in the environment. So seed is the basis of our agriculture. So in agriculture, seed is the basic thing. Farmers used to grow the plants. At the time of harvest, they use some amount of seeds for commercial purpose, some amount of seeds for their personal use, that means as a source of food, and some amount of seeds will be stored for the next year to produce a new crop. Okay, dehydration and dormancy of mature seeds are crucial for storage of seeds which can be used as a food throughout the year and also raise crops in the next generation. Imagine if the plants are all like tomato. In the case of tomato, you cannot dehydrate the seeds. If you want to grow the tomato, you just have to cut open the tomato fruit and place the seeds directly in the soil. It will grow into a new plant. You cannot dehydrate it. It requires a lot of moisture. But in some plants like rice, they only retain 10 to 15% of moisture, so which makes sure that they will survive in the unfavorable condition, and onset of favorable condition they can be used for the production of new crops. So can you imagine agriculture in the absence of seeds? We cannot imagine, but there are possibilities. Okay. Presence of seeds which germinate straight away soon after formation cannot be stored. How long do the seeds remain alive after they are dispersed? This period again varies greatly. That means in plant to plant, this condition, that means dormancy, viability, dehydration will be from plant to plant. There are some examples here. The oldest is that the Lupinus arcticus, excavated from Arctic tundra. Okay, so this plant, the Lupinus arcticus, seeds germinated and flowered after estimated of 10,000 years. Surprising, really surprising. Okay, this Lupinus arcticus plant seed retained its dormancy and viability for 10,000 years. Now tell me whether tomato seed can be stored this amount of or this duration of time. It is not possible. So it varies from plant to plant. One more example, there is a recent record of 2,000-year-old viable seed is of the date palm, that is Phoenix dactylifera, discovered during the archaeological excavation at King Harold's palace near the Dead Sea. Again, they got one seed of date palm which is around 2,000-year-old and it is viable. They produced the plant from those seeds. These are the two extreme examples where seed can be viable even up to 10,000 years. Now, in NCERT textbook, they asked some of the questions. We will try to answer those questions. Okay. How many eggs are present in an embryo sac? How many eggs are present? Only single egg. How many embryo sac are present in an ovule? Single embryo sac. How many ovules are present in an ovary? This will vary. It will vary greatly from a plant to plant. How many ovaries are present in a typical flower? This will also vary. Sometimes monocarpellary, bicarpellary, tricarpellary, some multicarpellary conditions are observed. Then how many flowers are present in a tree? Sometimes we observe a tree with full of flowers, thousands of flowers. Sometimes a tree is producing single flower in its lifetime. So like that, there are so many variations we observe in the number of flowers. Now, some more exceptions or interesting characters are given. Orchid fruits are one such category, and each fruit contains thousands of tiny seeds. That means if you observe orchid gynoecium or ovary, how many ovules may present? Thousands of ovules are present. After fertilization, all those ovules develop into seeds, That's why in the fruits we observe thousands of seeds. If possible, you just observe the vanilla pod. Inside that vanilla pod, thousands of seeds, tiny seeds are present. Okay. There are two more examples, that is Orobrac and Striga. Here I have given the Orobrac and Striga. You can see the plants. If you observe their fruits, inside that it produces thousands of seeds. They are parasitic plants. Okay. So in examination, they may ask this example as a parasitic plant. Have you seen tiny seeds of ficus? Ficus plant, have you seen? In that it produces so many fruits, and frugivorous birds, they used to come and visit the plants at the time of fruiting. And if you cut open that fruit and observe, seeds are very minute. Okay, but thousands of seeds are present. Then how large is the tree of ficus developed from that tiny seed? That is the beauty of nature. See, that tiny seed has genetic information. It can produce a tree which is of a size of an acre. Right? So how many billions of seed does each ficus tree produce? Even though they produce billions of seeds, possibility of producing offsprings will be very, 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 very less. From that billion seeds, it can produce 1 or 2 offsprings. Okay. Can you imagine any other example in which such tiny structures can produce such a large biomass over the years? We are asking some other example which is similar to ficus plant. Okay, if you want, you can search such type of plants in nature. Now the last part of the topic, and very interesting part of the topic, Apomixis and Polyembryony. So far we studied that even without the fertilization, ovary can develop into fruit, and that process we call it as parthenocarpy. Now just observe here, although seeds in general are the products of fertilization, a few flowering plants such as some species of Asteraceae and grass have developed a special mechanism to produce seeds without fertilization called apomixis. This is very, very important example. In the case of Asteraceae and grass, seeds are produced without fertilization. We studied production of fruits without fertilization, but here seeds are produced without fertilization, and that process we call it as apomixis. Now tell me, if the seeds are produced without fertilization, is this an advantageous process for us? Maybe for plants, some of the plants, it is a type of adaptation for survival. For us, it's really a boon, because apomictic seeds, if they are available, they are just like clones. The offspring produced are exactly similar to their parents. So scientists are very curious about how these apomictic seeds are produced in Asteraceae family and in grass. They are trying to introduce these apomictic seed production in other plants also, so that any farmer can get benefit from it. There is no need to visit the seed bank or any of the commercial scale production of seeds every year to grow the plants. Other than that, he can collect the seed from his own field and retain its hybrid character for many generations. Actually, what happens, once he collects the seed from the hybrid seed banks and sow it in the field, because of cross pollination, all the genetic compositions will be changed, and in the next generation, if we take the seed from the same plant and sow it in the field, there is a high chance that he loses his hybrid vigor. So introduction of apomictic seeds in the agriculture may be profitable for farmers. They can use the seeds produced in their own farm and use it for next generation production. Okay, so it has a great agricultural application importance. The fruit production without fertilization is called parthenocarpy. Apomixis is a form of asexual reproduction that mimics the sexual reproduction. Such a beautiful word. Apomixis is a type of asexual reproduction. Actually, seeds are produced without fertilization, right? It is exactly like a clone, but still which mimics the sexual mode of reproduction. It produces the seed. Seed production is only observed in sexual mode of reproduction. So there are several ways of development of apomictic seeds. in some species, diploid egg cell is formed without reduction, division, and develops into an embryo without fertilization. Okay, so more often, as in many citrus and mango varieties, some of the nucellar cells surrounding the embryo sac start dividing, protruding into an embryo sac and develops into embryo. And such species, each ovule containing many embryos, and that is called polyembryony. What happens here actually, inside the ovule, one embryo sac will be there, one egg will be there. After fertilization, zygote will be formed, zygote will develop into embryo. But here in this case of citrus and mango, what happens? Nucellar cells also becomes embryos. Okay, that means from one seed you can observe multiple embryos. That condition is called polyembryony. And occurrence of more than one embryo in the seed is referred to as polyembryony. What would be the genetic nature of apomictic embryos? Genetic nature, they are clones. Can they be called clones? Of course, yes, they are clones. Now, what is its application in agriculture? We'll try to understand. Hybrid varieties of several of our food and vegetable crops are being extensively cultivated, and cultivation of hybrids has tremendously increased the productivity. When the productivity is very high, usually farmers, what they will do, every time they go to a shop where they sell the hybrid seeds. So the cost of the hybrid seeds will be very high. See, in agriculture, if they spend more on hybrid seeds or chemical fertilizers or for labors, then the profit what they get at the end will be very, very less. So if this continues, most of the farmers, they lose their interest in agriculture, right? So in order to overcome this problem, one of the problem of hybrid is that hybrid seeds have to be produced every year. Because of cross-pollination, in the next year itself, it loses its hybrid vigor. If the seed collected from the hybrids are sown, the plant in the progeny will segregate and do not maintain the hybrid character. Because of segregation, what happens? The hybrid character cannot be retained. Production of hybrid seeds is costly, and hence the cost of hybrid seed become too expensive for farmers, as I said. If these hybrids are made into a apomictic, there is no segregation of character in the hybrid progeny, then the farmers can keep on using the hybrid seeds to raise the crops year after year, and he does not have to buy hybrid seeds every year. So this will be a great advantage for the farmers. No need to depend on hybrid seeds. They can use the seeds produced in their own crop field. Okay, because of this importance of apomixis, the hybrid seed industry, active research is going on in many laboratories around the world to understand the genetics of apomixis and to transfer apomictic genes into the hybrid varieties. If this got achieved, then the farmers will be rewarded better. Their profit ratio will be increased. Okay, so with this, we completed the explanation part of the topic sexual reproduction in flowering plants. We studied in detail about the pre-fertilization structure and events, then fertilization and post-fertilization and structure and events. Under that, endosperm, embryo, seed, fruit, and some of the exceptions like apomixis and polyembryony. Let us discuss some of the questions which appeared in the competitive exam like NEET. Okay,